Bismillah ar-Rahman ar-Rahim. Today is our lecture about physiology of hepatobiliary secretion, which is we will include liver, gallbladder, the ducts, the secretion of the liver, functions of liver, and we will see how the liver is playing role in digestion of fat and metabolism. Okay. Why the slides are not moving? Okay. So these are our objectives for today's lecture. In today's lecture, we will be discussing the functional anatomy of the liver. We will discuss the anatomy, but not the actual anatomy lecture, but it is the functional anatomy, how the liver functions and what are the parts important for physiological importance of liver as well as the biliary system. Then we will see the formation of bile, how the bile formed and how it is secreted. What are the contents of the bile and what is mean by enterohepatic circulation. Circulation means coming and going in circle of bile salts. How the bile salts are circulated between enteron and hepatic or the liver and the small intestine. Also, we will discuss the functions and component of the bile in terms of digestion and absorption of dietary lipids. How the lipids are digested with the help of bile. These are our objectives and how the control or the bile secretion is controlled by neural, hormonal, or chemical methods. How it is regulated, secretion is regulated. So as you know, liver is the most important metabolic factory of our body. It is called as the biochemical factory. All the metabolism is going on in the liver. Which metabolism? Metabolism of carbohydrate, metabolism of fat, metabolism of protein. All three major food components are metabolized. Along with it, vitamins, minerals are also metabolized or converted into its absorbable form or the actual form in the liver. So liver is, you can say it is a factory where metabolism is going on, biochemical reactions are going on. And it has got importance in mainly importance in the fat digestion of fat, how the fat is digested and absorbed also. We will discuss uh, the bile formation also, how the bile is formed. You know, the liver is the organ which is forming bile. The bile is secreted from liver. And once it comes out by the right and left hepatic duct, it will be stored in gallbladder. It will be stored in gallbladder here. And then during the time of food, when food is entering in the second part of the duodenum, it will be poured. It will be poured when the food comes from the stomach to the duodenum. Second part of the duodenum, at that time, this bile will be mixing with the food for the digestion. But in between, it is stored in gallbladder. So in total, biliary system means liver, the ducts, and gallbladder. Liver, ducts, and gallbladder together, they are known as biliary system. It is known as biliary system. Let us talk about what are the functions. There are so many functions of liver are there, but to remember, we have to divide them into categories. So major categories are two. Liver functions are divided into two main categories. One is called as the digestive function and another one is known as the non-digestive function. Non-digestive function. So two major categories of the liver function are digestive and non-digestive. In digestive function, mainly the fat digestion. mainly the digestion of fat. In the non-digestive function, there are metabolism, mainly the metabolism. Metabolism of what? All food, protein, carbohydrate, and fats, as it is written here. Also, it is important in detoxifying. Detoxifying. What is mean by detoxifying? Detoxification means what? Anything in our body when we eat, there may be some useful substance and some non-useful or toxic substance enter into the body, or they may be formed into the body during the process of metabolism. So if these toxic substances are not neutralized, then they may cause problem in our body or dysfunction of various organs in the body. And these toxic substance has to be neutralized, and it is neutralized with the help of one of the function of the liver is to detoxify or degrade them, break down these toxic substance into waste products, and then it will be removed from the body. So these toxic substance can be any chemical in the form of drug, in the form of end product of the hormone, or maybe any food additives. They are all toxic to the body, and it has to be removed or neutralized with the help of liver. So liver is helping in detoxifying the foreign substance or maybe the substance which are formed inside the body. Also, it has got functions related to synthesis. What it synthesize? It synthesize proteins, plasma proteins. You have heard about plasma proteins in heme immune block. Albumin, globulin, fibrinogen, these are major, but even a smaller amount of or different types of plasma proteins are synthesized in liver. So liver is a factory for synthesis of plasma protein. It has got also functions related to storage. What it will store? It will store glycogen, excess of glucose. If any person taken in food, then it will be converted into glycogen. It will be stored in liver. It can store fat. It can store iron, because iron is a very little amount is required. Suppose the person is taking more than the required amount of iron in the food, then it will be stored in liver. Also copper. Copper is also stored in the liver, and many vitamins. Many vitamins are stored in liver. Vitamin B complex. This is the major vitamin. This is stored in liver. It also secrete hormone. Not only store, it secretes some of the hormone. So liver is known as major, or it is the largest gland of our body. It is having secretory function. That's why it is also called as a gland. What it will secrete? It will secrete hormone like thrombopoietin. What is thrombopoietin? The hormone responsible for platelet formation. Hepcidin, the hormone responsible for regulation of absorption of iron. Insulin like growth factor, IGF. This is secreted by the liver. It also has got function of activation. Activation of what? Activation of vitamin D. Vitamin D. It starts forming in the skin. When sun rays fall on the skin, there will be pre-vitamin. Pre-vitamin is formed. Pre-vitamin. Then it will go to liver for hydroxylation reaction. Then there will be inactive or D3 vitamin D3 is formed, and then it will reach to kidney, where the actual activated vitamin D will be formed. Actual vitamin D is formed. So activation of vitamin D3, one step is taking place in liver. It also removes bacteria from bacteria as well as the worn out RBCs, the RBCs which has already aged, those which becomes older than 120 days or maybe more. When they are broken down, it will be removed from the body or from the circulation by the liver. As well as bacteria, some of the bacteria in the circulation will be removed by liver. It also has got excretory function. What it will excrete? It will excrete cholesterol and bilirubin. Remember, this is very, very important. Bilirubin is excreted by the liver. Why one should know all these functions? Why one should know all these functions? So activation, then removal of the bacteria or worn out cell, and excretion. These are all functions of liver. Why we should know? Because if suppose a person is suffering from any liver disease, suppose liver is damaged, not functioning, then there will be deficiency of all these things which the liver is performing. There will be deficiency of vitamin D. There will be deficiency of the stored vitamins. There will be deficiency of hormones, thrombopoietin or other hormones. There will be deficient or improper digestion and absorption or metabolism of various food elements. All this will happen if the liver function is improper or liver is damaged or suffering from a person is suffering from disease, liver disease. So that's why we have to remember what are the functions of liver. So basically liver performs two types of function, digestive function and non-digestive function. In digestive function, the digestion of fat. In non-digestive function, metabolic function, detoxification, synthesis, and storage, secretion, activation, removal of the bacteria and worn out RBCs, and excretion of the bilirubin and cholesterol. So these are all functions of liver. Please remember all of them. Then, as you know, liver is the main organ or the organ from where the bile is secreted or formed. The bile is formed in liver. What is bile? Bile is nothing but a liquid which consists of bile salts, cholesterol, lecithin, and bilirubin. All combined, it is known as the bile plus water. Plus liquid means it has got water plus all these elements. This is formed by the liver, but it will be coming out of the liver through duct. This is the liver. It will give out bile through right and left hepatic duct, which will come out as hepatic duct, and then it will be joined by the duct from the gallbladder, cystic duct. When cystic duct plus hepatic duct is formed, this will lead to common bile duct, CBD, common bile duct, and then it will open in the second part of the duodenum along with pancreatic duct. Along with the pancreatic duct, this will be opening here, which is guarded by a valve, here. Okay, so here is the pancreatic duct. Together, combined with bile duct plus pancreatic duct, it will open into the second part of the duodenum. And as it is secreting bile, it will be continuously secreted. It is continuously secreted, come out. And if this valve is open, it will enter into the small intestine or the second part of the duodenum. But if the person is not taking meal, because this valve is closed in between meal, when the person is not eating anything, this valve is closed. So where this bile will go? This bile will go back into storage, which is in the gallbladder. In gallbladder, the bile will be stored. This liquid bile consists of bile salts. Main constituent is bile salts. What are bile salts? Bile salts are nothing but these are the derivatives from the cholesterol. So basically it is formed by cholesterol. So basic element in the bile salt is cholesterol. It converts. What is the function of bile salt? It converts the big fat globule into smaller molecules. When bile salts act on the fat globule, it will be converted into smaller molecules which can be easily absorbed, or it can be acted by the pancreatic lipase. Pancreatic lipase will be acting on it for digestion, for metabolism. Okay, so these smaller particles will be formed by the action of bile salts. Basically, it is performing a reaction which is called as emulsion, formation of liquid emulsion. What is emulsion? Emulsion is nothing but if you take in a bottle, suppose this is a bottle, you take oil and you add water to it. When water is added to it, then there will be two layers will form, right? They will not mix with each other because these are immiscible, cannot be mixed. But if you add some soap to it, liquid soap or any detergent, if you add them and then shake it, then it will become soluble. The oil will mix with water. This is called as emulsion reaction. It will convert big fat molecules into smaller fat molecule. This is called as emulsification. And this will be helping to reabsorption into the blood. These fat molecules can be easily reabsorbed. These are known as micelles formation, micelle formation. These small particles are known as micelle formation or micellar formation. So this is the action of bile salts on the fat. So liver has got two types of cells. One is known as hepatocyte. The cells of the liver are known as hepatocyte. Plus there are other cells which are known as Kupffer cells. Two types of cells are present in the liver. One is hepatocyte, the one which is doing all the formation of bile, and second one is the Kupffer cells. All these actions, all the functions which we have mentioned, is done by hepatocyte, except phagocytosis. Except phagocytosis. These Kupffer cells are responsible for phagocytosis. Phagocytosis is the only function which is done by Kupffer cells. Rest all other functions are done by the hepatocyte, like digestive function, metabolic function, storage function, detoxification function, secretory function. All these functions are done by the hepatocyte. Kupffer cells are nothing but the macrophage cell. Kupffer cells are nothing but macrophage cells. So for functioning of liver should receive blood. So from where the blood supply to the liver comes? To the liver, blood supply comes from two main sources. What are these two main sources? One is from hepatic artery. Simple. Any artery or usually the blood supply to any organ comes from artery. So hepatic artery is the one which will supply the blood. Plus it also receive blood from the portal vein. Portal vein. Now, as the name suggests, it is a vein. It should carry the blood away from the organ, but here it is portal vein. It is bringing the blood to the liver. It is coming from where? Coming from small intestine. So from the small intestine, the blood which is or the food particle which are digested and absorbed in the blood in the small intestine into the portal circulation, and it will carry the blood to the liver. So liver is receiving blood from artery as well as from portal vein. Portal vein supplies around 75% of the blood, whereas arterial supply, which is about for the oxygen, it is about 25% only. Okay. So these are the two main sources of the blood. Once the vein enters into the organ or enter into the liver, it will be divided into capillaries. You know, any arterial enter into the organ, it will be divided into capillaries. Arterial will become capillary like this. So here the vein will break down into form the capillary, and the capillary in the liver is known as sinusoid. The name is a special name is given for the capillaries in the blood. Capillaries in the liver is known as sinusoid. Sinusoid. So this picture shows you how the blood flow is reaching to liver. Liver is receiving blood from hepatic artery, number one, 25% of the blood, and 75% of the blood is coming from the portal vein, this vein, portal vein, hepatic portal vein. And then it will be drained into the hepatic vein, which will join to the inferior vena cava and reaches to the heart. So to study the function of how the liver functions, we have to study a small part of the organ. As you are studying like functions of muscle, you study which small area in muscle. If you want to know the function of muscle, you study sarcomere, right? So similarly, for studying the liver function, you have to study a area known as hepatic lobule. Hepatic lobule. This hepatic lobule is nothing but a hexagonal structure. Hexagon means 6 sides, 6 corners. 1, 2, 3, 4, 5, and 6 corners are there. Hexagonal structure. In the center, it has got a central vein, and at each corner, it has got 3 vessels. 1, 2, and 3. Out of these 3 vessels, one is a branch of artery, hepatic artery. Another one is a branch of portal vein. Let me change the color. Where is my cursor? So another branch, another vessel is a portal vein. Portal vein. And the third one is, sorry, third one is a branch of bile duct. Where is my cursor here? So bile duct. Hepatic artery, portal vein, and bile duct. All these 3 are together known as portal triad. Portal triad. Portal triad. So similarly, this is the one structure. Second one will be from here. Second lobule will be from this side. So like this, central vein and the 6 sides will join to form like a honeycomb structure. Like a honeycomb structure. This is called as a hepatic lobule. Hepatic lobule. And from the vein, the capillaries will start and it will form the sinusoids. From the capillary, it will broken down to from the vein, capillaries will be formed, and these capillaries in the liver is known as sinusoids. So all these are sinusoids. Sorry, sinusoids. So blood from hepatic artery will be moving from periphery to the center. Blood from portal vein will be moving from periphery to center. And the bile which is formed will be moving from center to towards the bile duct to the periphery. This is how it will be moving. This is the same structure which is in the 3D formation. It is shown at every corner. At every corner, you have got a 3 vessels: hepatic artery, a portal vein, and a bile duct, branch of bile duct. And then they are moving into the center, which is called as which is forming the capillaries, which are known as sinusoids, and they are draining into the central vein. Like this, here also it is shown. So hepatic artery blood is showing blood is flowing from periphery to the center. Portal vein blood is flowing from periphery to the center, and the bile is moving towards the bile duct, towards the bile duct. Same picture is shown here. What I was telling you, that from the portal vein, it will be converted into sinusoids. So these are gaps. So bigger gaps are seen, and these gaps or these capillaries are known as sinusoids. Green color is the bile duct, branch of the bile duct. So in simple diagram, it is shown two types of cells are present in the liver. One is hepatocyte, this pink color, and another one is this green color. These are known as the Kupffer cells. So these are the Kupffer cells which are responsible for phagocytosis. Phagocytosis of what? Phagocytosis of bacteria, any viruses, or any worn out RBCs. RBCs which are older enough and they will be engulfed by the Kupffer cells. Rest all other functions are done by the hepatocyte. This is the structure which I was telling you, that there is a hepatic lobule, which is classical corner six hexagonal structure with having a portal triad at each corner and a central vein. If you join three central veins, you will make another structure which is called as portal lobule. So these are some of the histological picture of liver, hepatocyte. Liver blood flow, Kupffer cells are lining the sinusoids. So if any material is found in the blood moving, which is not useful for the body or any foreign bodies or any bacteria, they will be engulfed by the Kupffer cells because they are lining the sinusoids. So bile is continuously formed. Bile is continuously formed and secreted by the liver cell or hepatocyte. Then it will go to the bile duct. From the bile duct of different lobules, it will come out of the liver as through the right and left hepatic duct. They will combine along with a duct coming from the gallbladder, which is known as cystic duct, to form the common bile duct. Why I am repeating this common bile duct? Why? Because there are chances that it may be blocked. Common bile duct can be blocked by any tumor, adenoma, or by stone, which are stone which is formed in the gallbladder. So gallstones can block the common bile duct, and then it will lead to severe pain for the patient. Patient will suffer from having severe abdominal pain, which is due to the blockage of common bile duct due to stones. Then it will open into the duodenum. This common bile duct will open into the duodenum along with the pancreatic duct, along with pancreatic duct. Here is the picture. So right and left, this is right and this is left hepatic duct. Then this is the common hepatic duct. Then cystic duct is this one. This is coming from the gallbladder. And then cystic duct plus hepatic duct will form the common bile duct, which can be blocked, and it will open along with the pancreatic duct, this one, pancreatic duct. So this is guarded by a valve, which is known as sphincter of OD. Sphincter of OD. Maybe written here. Hepatopancreatic sphincter or sphincter of OD. Or maybe ODI. So this sphincter is remain always closed except during meal. Whenever the person is eating or food is entering into the small intestine, that time only the sphincter will be relaxed and secretion from the or bile will enter into the small intestine, as well as the pancreatic lipase will pancreatic secretion will also enter into the small intestine. So during when the person is not taking meal, this secreted bile will be stored into the gallbladder. So function of gallbladder is to store the bile for future secretion, and it may concentrate. It will concentrate, and the ducts are Opening into the small intestine along, it is guarded by a sphincter, which is known as sphincter of OD, which is closed all the time. So in between the meals, the bile will be stored in gallbladder. So in between the meal, gallbladder will be storing as well as concentrating. It will absorb water. It will remove water from the bile and make it concentrated bile. So around 500 ml of the bile is secreted per day. Every day, 500 ml of the bile is secreted by the liver, and its pH is alkaline in nature. Alkaline means pH is more than 7. pH is 8 because it contains soda bicarb. It contains bile salts. Main content of the bile is water plus H2O plus bile salt, some cholesterol, lecithin, phospholipid, and bilirubin. Bilirubin is which is derived by the hepatocyte activity. So these are the constituent of the bile. Water plus bile salts plus cholesterol plus bilirubin. Bile will not contain any enzyme. It is devoid of any enzyme. So though no enzymes are there, how the bile is functioning for the digestion of fat is by physical purpose. It will just cut down the bigger fat molecule into smaller fat molecules with the help of a process known as emulsification. Just like as if you are cleaning your hands after taking a meal, when your hands will become oily, that time you use soap and it will be removing the oil from your hand. And this process is known as emulsification. Similarly, in our body, the fat molecule, which is bigger enough, it will be broken down into smaller molecules with the help of these smaller molecules with the help of an action called as emulsification, which is done by bile. So after doing its function, let's say this is a duodenum. So the bile is secreted here and it will help in emulsification. This is a small intestine, and then this small intestine will open into cecum and appendix. Cecum, and then it will start the as a large intestine, right? So at this point, the lowest part, this part, by this time, the bile salts will be acting and they will be digesting the fat. Till here, it will digest almost all of the fat. Then it will be reabsorbed from here. In the last one third of the small intestine, all these bile salts which perform their function will be reabsorbed into the blood through the portal circulation and goes back to the liver. It comes from the liver here secreted into the second part of the duodenum, and after performing its function, 95% of these bile salts will be reabsorbed and goes back to the liver. This is known as enterohepatic circulation. Enterohepatic circulation. Circulation between liver and enteron. Enteron is a small intestine. So between the intestine and the liver, the biles are circulating. This is called as enterohepatic circulation. So almost 95% of the bile salts are reabsorbed in the terminal ileum, in the terminal small intestine or terminal ileum. Only 5% will be lost into the feces. Remaining 5% will be lost into the feces. This 5% will be again secreted by or formed by the liver cell or hepatocyte. So this is the meaning of enterohepatic circulation. Now the question arises, what will happen if there is problem in the terminal ileum and the doctor is removing the terminal ileum part? Suppose this is the terminal ileum. It is removed because of some tumor, because of some blockage. If it is cut by the surgeon, this area, then what will happen? There will be loss of or no reabsorption of bile salts, and this will be lost into the circulation, into the feces. Okay. This diagram shows you how the enterohepatic circulation is occurring. Bile salts are secreted by hepatocyte, reaching to the small intestine, performing its function, and by the time it reaches to the terminal ileum, 95% will be reabsorbed. And after reabsorption, it goes back to the liver. This is called as enterohepatic circulation. So on an average, this cycle of enterohepatic circulation, meaning how many times during each meal, the cycle will go on twice. One time it is secreted and reabsorbed. Second time it is secreted, second time, and then reabsorbed. So two cycles are going on in one meal. And out of that, 95% will be restored, whereas 5% will be lost into the feces. So basically, there is no reaction, no chemical reaction is happening between bile salts and the fat. It is the physical reaction. We call it as emulsification, just like breaking down of fat molecule into smaller fat molecules, which is known as micellar formation, so that these small molecules will be available for the action of pancreatic lipase. Pancreatic lipase can act on these smaller molecules. So if no bile salts are present, then it will take long time for the lipase to do the fat digestion. Fat digestion is actually happening because of the pancreatic lipase. And if the bigger globule is there, molecule is there for the fat, this pancreatic lipase will not be able to perform its actual function or the function in total. This is the diagram showing you. This is the blue and yellow color. These are the bile salts which are attaching to a big fat molecules. So large fat molecules will be converted into smaller, smaller, and further smaller. This is called as liquid emulsion, formation of smaller fat globules. It is known as emulsion. It can be easily absorbed or it can be easily acted by the pancreatic lipase. So as I was telling you, the smaller particles are known as of the fat are known as the micelles. They are known as micellar formation. These micelles are about 3 to 10 nanometer size, very small as compared to the emulsified fat droplets, which is 100 nanometer. Oh, sorry, 1000 nanometer. Bile salts, cholesterol, and lecithin will help in micelle formation. Micelle will carry monoglyceride. It will be having monoglyceride, not the fatty acid, not the triglycerides or the bigger fat molecules. It contains monoglyceride as well as fatty acid. Okay, so as we know that bile is stored in gallbladder. Bile is stored in gallbladder. While stored, it will become concentrated. Water is reabsorbed. Water is reabsorbed, and it will become very concentrated here. It may lead to formation of gallstones. It may lead to formation of gallstones because it is a salt. So when salt loses water, it will become crystalline, and this crystal will combine to form the stones. So these stones are maybe out of that these stones, 75% of the gallstones are because of the excessive cholesterol, or it is formed by the cholesterol, and 25% is due to bilirubin, due to bilirubin. So gallstone are of two types: cholesterol stone or bilirubin stone. Bilirubin stone is 25% and cholesterol stone are 75%. What is bilirubin? Bilirubin is a end product or waste product formed in our body. Waste product formed in our body, and it is excreted in bile. It is excreted in bile and it will come into the feces. From where it comes? Bilirubin comes from when RBC broken down. RBC contain hemoglobin, right? So when RBC ruptures, hemoglobin will come out. It will give you two parts. One is heme part, one will be heme, and second one will be globin. Globin will be used for synthesis of new globin or new plasma protein. But heme part will go under many steps of reaction to form the bilirubin, which is a waste product. So it will not be reabsorbed, it will be removed from the body and it is excreted by the liver cells. So bilirubin is an end product of hemoglobin metabolism. And liver cells, they are excreting it, removing it from the body continuously. If liver cells are not functioning properly, what will happen? Bilirubin will remain into the body and its concentration will increase. Its concentration will increase. As bilirubin is a yellow color pigment, bilirubin is also known as a bile pigment. It is a yellow in color. So if it increases in our body, the body color or various uh, you can say mucosal surface will become initially yellow in color. This is known as jaundice. Jaundice is nothing but increased bilirubin concentration in our body. Normally, the bilirubin concentration is less than 1 milligram per 100 ml or percentage per deciliter in our body, less than 1 milligram. If the concentration increases, maybe it increases to more than 3 milligram, then you can easily see the yellow discoloration of sclera. Yellow discoloration of the sclera is seen. In the eyes, suppose this is an eye. So this white part, outer part, this white outer part will show you yellowish discoloration. This is known as due to increased bilirubin concentration, and this is seen in condition known as jaundice. So this diagram shows you how the bilirubin is formed in our body. Hemoglobin from the worn out cells or ruptured RBCs will be come out as heme and globin. Hemoglobin will be converted into heme plus globin. Hemoglobin will lead to formation of bilirubin, which is unconjugated. Bilirubin, free bilirubin, you can say. Bilirubin is free without any attachment here. This is called as unconjugated bilirubin. In the liver, it will be combined with some chemicals. This is called as conjugation reaction so that it will not harm our body. And then it will be released into the small intestine and through feces as stercobilin it is removed. Through kidney, it is also removed as urobilinogen. So yellow color of the urine is because of urobilin. Yellow color of urine. And also the brown or yellow color of fat, brown or yellow color of fat is because of the bilirubin, that is stercobilin. Here it is known as stercobilin. In the intestine, it is acted by bilirubin is acted by the bacteria to convert it into urobilinogen and finally stercobilin, which gives the brown color of the feces. If the bile duct is obstructed, as I was telling you before, if the bile duct is obstructed by stone, then color of the feces will be turned into gray or white. Clay color stool will be formed. This is because of the obstruction to the bile duct so that bilirubin is not reaching to the small intestine. Okay. In the intestine, it will be reabsorbed and excreted through the urine as urobilin. Normal concentration of bilirubin is 0.3 to 1.2 milligram per deciliter, normal concentration. If it increases in our body, the condition is known as jaundice. Now you can tell me what are the causes of jaundice? Causes of jaundice. Causes of jaundice are classified as causes before the liver. This is called as pre-hepatic jaundice. Causes may be related to liver, that is hepatic jaundice. And it can be related to the obstruction of the duct. This is called as post-hepatic jaundice. Post-hepatic jaundice. So three types of jaundices are there. All these conditions will have yellow discoloration, but the causes can be classified as causes or etiology before the liver. This is called as pre-hepatic jaundice. Pre-hepatic because of excessive hemolysis, sickle cell anemia, or thalassemia. Any cause hepalysis or mismatched transfusion of the blood can lead to excessive hemolysis. So, lot of bilirubin is formed, and liver has no capacity to conjugate it. Liver has no capacity to conjugate. So, unconjugated or indirect bilirubin will be more in case of prehepatic jaundice. Unconjugated bilirubin will be more in prehepatic jaundice, hemolysis or any type of hemolysis. In hepatic and post-hepatic jaundice, usually conjugated bilirubin are more common. Increased conjugated bilirubin will be more. This type of jaundice, hepatic and post-hepatic. In hepatic, what are the causes? It can be inflammation, infection because of the viral or bacterial hepatitis. It can be due to cirrhosis. And post-hepatic, it could be due to blockage of this duct, either because of stone or because of any tumor, adenoma, can lead to post-hepatic jaundice. So please remember the types of jaundice: prehepatic, hepatic, and post-hepatic. And you can know which type of jaundice based on concentration of, first, increased concentration of bilirubin. First, there will be increased concentration of bilirubin, which is leading to jaundice. After that, you have to know the cause by knowing the concentration of conjugated or unconjugated bilirubin, which is higher. Okay, so bile secretion is controlled by, in GIT, these are the mainly three mechanisms, either due to mechanical regulation, chemical regulation, chemical regulation, or hormonal regulation, or neural regulation. Chemical regulation, the bile secretion will be most potent stimulus for bile, bile secretion is bile salts. Presence of bile salts will increase the bile secretion. Presence of bile salts or bile salts are the most important stimulus for bile secretion. So any substance which increases bile secretion, that substance is known as cholaretic substance. Cholaretic substance are those substances which increases bile secretion. And most potent is the bile salt. Hormonal regulation, secretin is the hormone. Secretin is the hormone which will increase or stimulate watery bile secretion, watery alkaline bile secretion. Neural mechanism, vagal stimulation will have very minor role in increasing the secretion of bile during the cephalic phase of digestion. When you are thinking about the food, at that time also some bile is secreted, but it's very minor, not major. So food in the duodenum, and mainly the fat in the duodenum, causes release of another hormone which is known as CCK, cholecystokinin. The full form is cholecystokinin. CCK hormone is released if the food or fat is present in the duodenum, which will cause contraction of the gallbladder, So the stored bile will come out into the small intestine. So whenever food enters into the small intestine, and mainly the fat, can lead to contraction of the gallbladder because of increased CCK secretion. So cholecystokinin is not increasing the secretion of bile, but it is causing the stored bile to come into the small intestine. The main hormone is secretin, directly stimulated by the secretin. So gallbladder is acting as a just storehouse, and it is concentrating. It is absorbing the water from the secreted bile or stored bile in between the meal, and it will make it concentrated. So the capacity of the gallbladder is around 50 ml. 50 ml of the bile can be stored in gallbladder, and if it is more, it will be just reabsorbing water and making it concentrated. So there may be in some conditions where there is a less water or it becomes very concentrated, then it may lead to formation of crystals and then gallstones. One interesting about the gallstone is very common in females. So female prevalence of gallstone is more than the male. And which female? Not all females. Those females which are fertile. That means they are in the age group of reproductive age group, fertile. And those who are obese. That means they are fatty. So obesity, female, fertile, and in the age of 40 years, 40 years, these are the four F's: female, fertile, fatty, 40 years is common for gallstone occurrence or incidence. Now in this lecture, you have to know the clinical conditions related to increased bilirubin concentration in the body, which will lead to a condition called as jaundice. As I told you before also, that yellowish discoloration will occur because bilirubin is yellow in color. And this yellow discoloration will be seen by the physician or by any person in a patient when the concentration becomes more than 3 milligram. At that time, the sclera of the eye will become clearly yellow. If it becomes around 8, then there will be facial yellowish discoloration will be seen. If it becomes more than 15, then whole body will look like yellowish. So as the concentration increases, the yellowish discoloration will go increases. Causes of jaundice, I told you, increased bilirubin concentration will be either due to prehepatic cause when the problem is before the liver. That means example is hemolysis, excessive hemolysis leading to excessive hemoglobin coming in circulation, which is converted into heme plus globin, and heme will go in various reactions and lead to formation of bilirubin. So if more RBCs are destroyed, more bilirubin is formed. This is cause does not related to the liver, so that's why it is called as prehepatic. There can be cause related to the conjugation where bilirubin is not get conjugated, or maybe the liver cells hepatocytes are not functioning, like in case of viral hepatitis A, B, C, D. All these type of hepatitis or cirrhosis can lead to increased bilirubin concentration, that is conjugated type. Conjugated bilirubin will be increases. Here unconjugated bilirubin will be more. Unconjugated. Unconjugated is also known as indirect bilirubin. Indirect bilirubin. Conjugated bilirubin. Conjugated bilirubin will be high in viral hepatitis or causes in hepatic jaundice. It is also known as direct bilirubin. There can be problem after the the bile enter into the duct, that is called as post-hepatic. That can be problem related to obstruction of the bile duct or anywhere in the path. It is called as obstructive jaundice. Most common is the stone in the bile duct, a stone blocking the path of the bile secretion. So in summary, what we have learned in this lecture, what are the different types of functions of liver, how the liver receives blood from where and where, two different types like hepatic artery and portal vein, how the bile is secreted, because main function of the liver is to secrete bile, which will help in digestion of the fat. What are the contents of the bile? Bile salts, how they are circulated, that is called as enterohepatic circulation. How is the bile acts on the lipid to cause the emulsification or digestion of the fat. Then what is bilirubin, its source and its concentration and the conditions related to it. How the bile is secretion is controlled, chemical, hormonal and neural. And we discussed jaundice in detail, that is increased bilirubin concentration in the blood more than 1.2 milligram, leading to yellowish discoloration. We call it as jaundice. Three types of jaundices are there. Cause before the liver, it is called as prehepatic jaundice, like sickle cell anemia or any hemolytic disease. There could be problem in the liver, that is hepatic jaundice. There could be blockage of the duct, it is called as post-hepatic jaundice. Did you get all these points?
Yes, doctor.
Thank you very much. So nobody other than Naji join our class.
Yes.
Naji, did you get the information properly now?
Yes, of course, doctor.
Was it easy?